We will continue from the previous lecture. We are discussing about the seminiferous tubule. In this photograph, a easy way of identifying this Sertoli cell is observing presence of cytoplasmic crystals, the crystals of Renke. All these black color spot you can see here, these are the crystals of Renke. That is a unique feature of human leading cells. These crystals are not stained with the common histological stain, but it can be recognized as a negative image in stain preparations. Now here in this very close magnified view of this electromicrograph trying to show us a Sertoli cell. Now here if you carefully observe this tail of a tail of that sperm, the body is attached to some stalk like structure here. Okay, so if you consider this as a stalk like structure, these all sperms are attached on this stalk. These stalks are a Sertoli cells. Okay, I hope it clearly visible here. This is a stalk portion and all the sperms are attached on the stalk. This is a Sertoli cell which provides nourishment to these sperms before they get released into the ejaculatory duct. This is a spermatogenesis, how the sperms are formed that from the primary spermatogonium. So that single spermatogonium goes on dividing meiosis and they reach to the sperm single mature spermatozoa. This is seminiferous epithelial is shown in this photograph. Now see here early developing spermatids are shown and these are the mature spermatozoa. So this is a spermatogonial epithelium. This is the one side of a seminiferous tubule, this is another side of seminiferous tubule. Now in this photograph you can clearly see this early spermatid development. This is a chromosomal structures are visible and you can see different dividing stages, meiotic stages in this, meiosis stages in this seminiferous tubule cross sections, and goes on in the developing and they form a late spermatid, mature spermatid remain attached to this stalk like portion, that is a this is here is a Sertoli cell. Now development of spermatozoa, just for a reference purpose, there are different phases of development of this spermatozoa, from the spermatogonium, Golgi phase, then acrosome phase, cap phase, maturation phase. This is a diagram of a mature diagnostic presentation of a mature spermatozoa with the acrosomal cap, head piece, then these are the middle piece, then all this tail portion and the end piece. They have shown the cross sections. Now hormonal control of this male reproductive system, there you will find functions of leading cells. Do you remember where we observe that leading cell? They are present in a surrounding gaps of seminiferous tubule. Leading cells are outside the seminiferous tubule and their role is to produce the testosterone. In response to this, interstitial cell stimulating hormone or growth hormone, these leading cells produce testosterone. Then the role of testosterone is in the development of seminiferous tubule, the proliferation and differentiation of spermatozoa, they form that seminivesicles, male accessory glands, and development of a secondary sex characteristics. All they are performed because of a secretion of these leading cells, which are in feedback mechanism. They get stimulated by the interstitial cell stimulating hormone from the pituitary. So here leading cells or interstitial cells of leading are shown in this photograph. This is the space. This is a one seminiferous tubule, another seminiferous tubule, third seminiferous tubule. In the gap of these three tubules, you will find a large nucleus with the cuboidal cells that produce this. These are the leading cells which produce these hormones. In this electromicrograph, again you can see a very large size of a nucleus and lot of endoplasmic reticulum in the cytoplasm of leading cells. Now we go to the uh, next stage of this seminiferous tubule. So once that spermatozoa or sperm is formed in these tubules, seminiferous tubules, they are poured into this portion. This portion is called as a retitesti, and the connection between retitesti and the sperm, that seminiferous tubule, is a tubule recti. So we'll see histology of this portion. So we must see this histology, because though it is not there in syllabus, but to separate exactly a seminiferous from other tube system in testes, we must learn this histology. So tubule recti is a straight tube, that is, and the transitions of only Sertoli cells to cuboidal cells, no cilia. Here in an epithelial layer, you will not find that spermatogonia thing in an epithelium, and you will not see any Sertoli cells, but you will see there the cuboidal cells. These ratatisti, they are located in the mediastinum, posterior thickening of a tunica albugia, and they forms a network of channels. And there you will find epithelium type is a cuboidal epithelium. So this is a ratatisti and efferent ducts. So efferent ducts, these efferent ducts are attached with the ratatisti tubule and the seminiferous tubule. So if we started from the seminiferous tubule, that is basic testis component. So all that sperms are poured through these channels, a large network channel, and that sperm travels and poured into this collecting duct, that is efferent duct. Now histologically you will see that here in a ductily efferentia, you will find these are the cuboidal cells lining. Epithelial lining is a cuboidal cell lining is there, and this retitestis, you'll find they have channels with a no Sertoli cell present in that. Now next tube system in a testis is after retitestis, it comes into the these epididymis portions. So we'll see the histology of epididymis now. Duct, that epididymis is called as a ductus epididymis. They are single highly coiled tube around 4 to 6 meter in length. They are kept in a very coiled form. It is supposed that during when the sperms are stored in this epididymis, they are get matured, they are ready to relieve from the ejaculatory ducts. So it is a 4 to 6 meter in length and kept in a very coiled form. This tubule plus surrounding connective tissue together form this epididymis. Head, body and tail region is present in case of this epididymis. So in a previous slide only we must have observed that label, that is quad epididymis, ducta epididymis, like things. So here they internally they are lined with the pseudostratified columnar epithelium, and naturally they have some ciliary type cells to promote the passage of sperms. Columnar cells are the principal cell in the epithelium. Basal cells are there, that is stem cells to replace columnar cells. Basic original cells from which the new columnar cells are developed. The surface of stereocilia. Stereocilia, they beat in a one direction only. That is what is a stereocilia. And so that epithelium is a pseudostratified ciliated columnar epithelium. Then after that epithelial layer, you will find there is a smooth muscle coat in case of a head and body region, and circular layers of muscles at the caudal portions. Now here they have shown the photographs of the epididymis and the efferent duct. Now carefully, if you say, try to identify different tubules here, this is a efferent duct and these are the epididymis. See the structure of the epithelium here, it is a pseudo-stratified columnar epithelium, and here it is a simple cuboidal epithelium is there, and tubes are small, irregular. Here it is a wide tubes, with lot of secretion over the surface. Here in diagrammatically, if you try to present the cross section of epididymis, so centrally all these are the spermatogonia, that is a mature sperm, spermatozoa. Forget about this, you can highlight it as a empty cavity, lumen. Then basal membrane, basal cell layer is there, basal membrane is there, and the principal cells are of which type? Pseudo stratified stereociliated epithelial, columnar epithelial cells are there. Okay, so here's the actual section where we can confirm these structures. So these are the nucleus. You see the arrangement of nucleus, which indicate that they are pseudo-stratified epithelial cells, and the border you will see some cilia. So it's a stereo ciliary epithelial cells is there, and the cavity will full with the spermatozoa. The functions of epididymis is what? They store sperm for maturations, breaking down non-ejaculated sperm. If there are some faulty sperms are there, if the sperms are broken or damaged, they get digested, broken down, and absorption of excess fluid secreted by the seminiferous take place in the epididymis, and propulsion during ejaculations. All that smooth muscles are there, and ciliated epithelial is there, which help in a propulsion of these mature sperms towards the ejaculatory duct, to the duct system for expelling out. Now again we come with the same testes diagram. Now we'll see the ductus differences, histology of these ductus differences. Now in next topic we'll see the ductus differences histology here. What is that ductus differences? It is a aka vasa difference. This term is a aka vasa difference. It is a straight tube and start at inferior pole of testis. That means at the end of the testis it start and ascend the spermatic cord. This enters abdominal cavity through the deep inguinal ring and enter the prostate at the area called as the ampulla and become the ejaculatory duct. So you can say this is a final outing tube of a testis. This is a tube which final tube which collect all the secretions from the testes and pour it into the ejaculatory duct, and this is aligned with the pseudostratified columnar epithelium. So same that epididymis epithelial structure continues with the ductus differences. There are three layers, thick smooth muscles wall is there. So slowly as we advance from epididymis to ductus differences, you will find that the muscular layer goes on increasing to increase the propulsion functions in that duct. One component of the spermatic cord is there along with the testicular artery, pampiniform plexus, and nerves are innervated to this duct, which helps in a functioning of this ejaculatory duct. Now, vasa difference, ductus differences, the histology you can see here, they are lined with the pseudostratified columnar epithelium, and you can see how much quantity of muscular layer increase here. Inner longitudinal muscle, middle circular muscles, outer longitudinal muscle. So you can observe the increased volume of muscles for providing the contraction, burst of a contraction, so that all the content present in a lumen should get propelled forward. The rest of the histological structure is same as the epididymis. They have pseudostratified columnar epithelial cells. Okay, in diagrammatic presentation, this is a all muscle portions, outer adventitia cord, then this inner longitudinal muscles, thereafter outer this intermediate circular muscles, then outer longitudinal muscles. Okay, now here we'll see some university exam papers questions from the question bank of a university exam paper of mammalian histology related with this testis topic. So they ask 1 mark questions like what are leading cells, I hope you can answer now. What is the functions of leading cells? We just discussed that is a secretion of hormones. What is the functions of Sertoli cells? It is a nourishment to the developing sperms. And what are the Sertoli cells? They are the columnar cells present in a seminiferous tubules. What are the cells of leadings? They again the cuboidal cells present in a gap between the seminiferous tubules, and they produce these hormones. State the functions of leading cells. Again the same questions asked repeatedly. Now some descriptive questions they ask in a university exam related with these topics are: describe histological structure of testes. Okay, here entire structure of testes has to be discussed. Major focus on seminiferous tubule, the outer layers, scrotum, then two that layers, and then septum and seminiferous tubule structure, then structure of that retitestes, epididymis, all has to be discussed under this heading of a entire histology structure of testes. Then sketch and label TS of testes. The one diagrammatic presentation just you have observed now, showing the entire testes in a with the all duct system that has to be drawn. Then describe the histological structure of seminiferous tubule. Now here you only draw a structure of seminiferous tubule. Now you don't have to draw entire TS of testes, but only the seminiferous tubules, and three seminiferous tubule adjusting three seminiferous tubule, and in between you show the presence of leading cells. That is required in the histological structure of seminiferous tubules, with all required that labels, basal membrane and spermatogonial epithelial layers and mature spermatid and all that things has to be described in the structure of seminiferous tubule. Thereafter write notes. Sometimes they ask for 4 marks, that is for the TS of testes. You draw that diagram, TS of testes, but what you will show in a TS of testes, majorly a seminiferous tubule cut into the sections, and some portions of epididymis or retitestes can be seen. So you show that diagram and you describe that portion in under that TS of testes. And in a TS of seminiferous tubule, again the same repeated question, like describe the histological structure of seminiferous tubule, that has to be repeated here.